Hello. Very good afternoon to the student who have joined. Can you please let me know if my voice is audible to you? Let me know if you can hear my voice. Okay, thanks. Thanks for confirming. Now please let me know if the presentation is visible to you. If the presentation is visible to you, kindly let me know. Okay, thanks for confirming. We will wait till more students join and then we will start today's session. Meanwhile, please wait. We will start the session shortly at 4 p.m. Let's wait till other students join. We will start the session at 4 p.m. So please wait, let other students join, and then we are going to start today's session. Okay, so let's start with the introductions. I think more students will join in due course of time, but it's already 4 p.m. So let me start by introducing myself. So, very good afternoon to the students who have joined. My name is Mousumi Deb, and I'm a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I'm your TA for this NPTEL course of Medicinal Chemistry, and today it is our week 9 live session. So, before starting the session, always I try to point out few things. There are few things which I need to summarize. The first is like there will be a problem solving session every week like on every Friday from 4 to 6 p.m. And these sessions are live on Google Meet platform. The link of the Friday session is this one. You can write it up on a place or you can save it somewhere and use the same link to join every Friday class from 4 p.m. And during the session, since the sessions are live, if any network disruption happens from my side, do not leave the session immediately. I will try to rejoin as soon as possible. Okay, and if there is any network issue from your side, the meet is open. The meeting is open, so you can rejoin at any time. So there is no issue with that. And since I'm recording the session, so that I can record the session without any issue, I have muted your microphones and cameras. Okay, but the chat box is always open, and always I monitor the chat box every now and then during the session also. So even if you have any doubt, any query, you have any issue with the microphone or camera or PPT, you can let me know through the chat box. Okay, the chat box is always open here. But since I'm recording the session, I have muted the microphones and also the cameras so that there is no issue in recording. After the class, if there is any doubt which was not answered during the class due to the short time, what you can do is you can drop your doubts through the email to me. I will share my email ID at the end of the session. The doubts can be from the today's class or any previous class or any basic question from the course of medicinal chemistry. You can just drop an email to me. And this presentation and the class recording, which I have already mentioned earlier. So these things will be shared to you soon. Okay. And where to find this thing, which I have already shown you a couple of times, that you have to go to the SWAYAM portal, and during the on the course announcement page, where there is an announcement for every week's course content release and the assignments and so on. So here there is the tab which is known as the problem solving session. You can go to that particular problem solving session recording, and when you enter this page, what we will do is, if you click on this thing, you will redirect to a page which contains the all the class recordings, and also the Google Drive link which contains all the previous class material, and all the materials in futures will be also uploaded to that particular Google Drive. Okay, so you can just go and download it from there. Okay, so today it is our week 9 live session. So what we do in every live session, we basically solve MCQ type of question, which are related to that particular week's course content. Okay, so since it is a week 9 live session, today what we will do is we will solve the MCQ questions or multiple. So this kind of questions all will be based on the week 9 course content, because week 9 is right now live, and you have to submit your week 9 assignment next. So today, what our all the questions will be based on the topics which are included in week 9. So what are these topics? Let us first check this out. So all the topics are basically related to the prodrugs. Okay, so optimizing the access to the target. So these things we have discussed already in our previous session. We have discussed that the optimization of the drug and target interaction. So little bit we will discuss it today also. Then we will go to the prodrug and drug alliances, endogenous compounds, peptidomimetics, and oligonucleotides as drugs, and optimizing the access of the prodrugs. Okay, so all these things which we will discuss today, and all our questions will be related to this course content. So let us start with today's session. I will show you the first question of today's session, and there will be four options: A, B, C, and D. You have to identify which of the following will be the correct answer. Okay, and after checking your answer, I will discuss that particular topic in detail. Okay, so let us first go to the question number one. So the problem number one of today's session is, it says, which of the following statements is false regarding prodrugs? Your options are: number A, metabolic enzymes can activate prodrugs. Number B, in general, prodrugs are inactive compounds. Number C, prodrugs cannot be activated by photodynamic therapy. And number D, not all prodrugs are activated by esterase enzyme. Which of the following statements is false regarding the prodrugs? I will give you some time. I will give you 1 or 2 minutes. Then I will check the chat box. After that, we will discuss that particular topic in detail. Which of the following statements is false regarding prodrugs? Number A, metabolic enzymes can activate prodrugs. Number B, in general, prodrugs are inactive compounds. Number C, prodrugs cannot be activated by photodynamic therapy. And number D, not all prodrugs are activated by esterase enzyme. Okay, let me check the chat box. Okay. Okay, I got answer C. Let me check if number C is the correct answer for this first problem. So the correct answer for problem number 1 will be yes, it is number C, which says that prodrugs cannot be activated by photodynamic therapy. So basically, what are prodrugs? So prodrugs are compounds which are inactive in themselves, but which are converted in the body to the active drug. They have been useful in tackling problems such as acid sensitivity, poor membrane permeability, drug toxicity, bad taste, and short duration of action. Usually, a metabolic enzyme is involved in converting the prodrug into the active drug, and so a good knowledge of the drug metabolism and the enzymes involved allows the medicinal chemist to design a suitable prodrug which turns drug metabolism into an advantage rather than a problem. Prodrugs have been designed to be activated by a variety of metabolic enzymes. Ester prodrugs, which are hydrolyzed by esterase enzymes, are particularly common, but prodrugs have also been designed which are activated by N-demethylation, decarboxylation, and the hydrolysis of amides and phosphates. Not all prodrugs are activated by metabolic enzymes. However, for example, photodynamic therapy involves the use of the external light source to activate the prodrugs. When designing prodrugs, it is important to ensure that the prodrug is effectively converted to the active drug once it has been absorbed to the blood supply, but it is also important to ensure that any groups which are clipped from the molecule are non-toxic. Okay, so in the most important thing which you have to remember regarding prodrugs is what is the need of the prodrug, what are the requirement, why we have to design prodrugs for some certain therapeutics. Okay, so the main reasons which I have mentioned here is that the acid sensitivity, some drugs are acid sensitive, so that's why we need to give the drug in a prodrug formation, so that there is no it can handle these things. There is a poor membrane permeability. It is also a very good reason. Then we have the drug toxicity. Sometimes the compounds have a certain toxic side group or something which can be tackled by prodrug designing. Bad taste, short reaction duration of action. These things, these are the problems why prodrugs are important. And as I have mentioned in the so the statements which if you notice the statement, the first one says that metabolic enzymes can activate prodrugs, which is correct. Then in general, products are inactive compounds, which is also correct, and not all prodrugs are activated by the esterase enzyme. It is also correct. Okay, there are many other ways by this prodrugs are activated, like N-demethylation, decarboxylation, and hydrolysis of amide and phosphate, and so on. Okay, and this statement number C, which says prodrugs cannot be activated by photodynamic therapy, this statement is false. Okay, because here it is clearly written that photodynamic therapy involves the use of an external light source to activate the prodrug. So this is statement is that's why now statement number C, which says that prodrugs cannot be activated by photodynamic therapy, it is false. So what are the advantages of prodrugs? So number one is an increase of water solubility by using sodium succinate esters or chloramphenicol in succinate IV injection. An increase of lipid solubility, for example, increase the duration of action by lipid soluble esters, and increase of the oral absorption as by esters, the highly polar groups or in methylation. Number six is the increase of topical absorption of steroids by masking OH group as the esters and acetonides. Then we have a decrease of water solubility to increase the palatability as in chloramphenicol palmitate. Then decrease in the GI irritation, side effects as aspirin. Then site specificity as in methyl dopa, and increased half-life and chemical stability as in the cefamandol acetate and a stable product, while the parent cefamandol is the unstable solid orange form. Hetacillin is another product which is for ampicillin. Okay. So these are the advantages of the prodrug, and this all about all the what is the necessity of prodrug and what is all the problems which can be tackled by prodrugs and so on. Okay. So now I will go to the next problem. Mhm. Okay. Now let us go to the next problem. So problem number 2 says which of the following will not be limitation, no, will not be a limitation of for prodrug design. Okay. Your options are number A, formation of toxic product. Number B, an inert carrier can be clipped off, forming toxic product. Number C, product cleaving off before reaching the target site. And number D, the cleaving of the carrier is also site specific. Which of the following will not be a limitation for prodrug design? Number one, formation of the toxic product. Number A, number B is the an inert carrier can be clipped off forming toxic product. Number C, product cleaving off before reaching the target site. Number D, the cleaving of the carrier is also site specific. Which of the following will not be a limitation for prodrug design? Again, I will wait for a couple of minutes. Then I will check the chat box and I will show you the correct answer and we will discuss it. Okay, let me check the chat box. The given answer is number B. Let me check if number B is the correct answer for problem number 2 or something else is the correct answer. So the correct answer will be it is number D. The cleaving of the carrier is also site specific. This statements it is not a limitation for the product design. Okay, this number D is the correct answer. So now what is prodrug and product design? I have already discussed it a little bit. So let us just rephrase it. So the prodrug is a pharmacological substance that administered with an inactive form. Once administered, the prodrug is metabolized in vivo into an active drug within the body through metabolic process such as the hydrolysis of an ester to form the drug. And sometimes the drugs are designed to make use of the metabolic processes in order to generate their active form. This is done in order to improve some selected property of the molecule such as the water solubility or ability to cross the membrane temporarily. What is the limitation of the product? So the problem associated with the product design is its toxicity, which is due to the formation of the unexpected metabolites from the total drug conjugates. Toxicity may be due to the inert carrier generated by the cleavage of the promoiety and the drug conjugate, which is converted to the toxic metabolite. The product might consume a vital cell constituent such as the glutathione during its activation stage, which causes the depletion of the prodrug. So the limitation of the product designing is the formation of the unexpected metabolite. An inert carrier can be clipped off, forming a toxic molecule, and the product might consume vital nutrients. If the carrier cleavage is site specific, it is an advantage rather than a limitation. Okay. So the correct answer for the problem number 2 was number D. Okay, because the site specificity uh, because the site specificity, it is the advantage, not the limitation for the product design. Okay, so correct answer will be problem number, no, sorry, the option number D was the correct answer for second problem. Okay, now let me go to the next problem of today's session. Okay, so problem number 3. It says that what sort of therapy involves the administration of an antibody linked to an enzyme such that the enzyme activates the prodrug? Your options are number A, ADAPT, number B, GDAPT, number C, ADEPT, and number D, GDEPT. What sort of therapy involves the administration of an antibody linked to an enzyme such that the enzyme activates the prodrug? ADAPT, GDAPT, ADEPT or GDEPT? A, B, C or D? Let me know in the chat box which of the following will be the correct answer for problem number 3. Even if your answer is wrong, it is fine because we are not going to mark your answers for the problem solving session. There is no mark, so you do not need to worry. There is marks in the assignments, that is another issue, but there is no marks answering during the question. It is for your practice only. So let me know A, B, C, or D, which of the following you think is the correct answer for problem number 3, which says that what sort of therapy involves the administration of an antibody linked to an enzyme such that the enzyme activates a prodrug? ADAPT, GDAPT, ADEPT, or GDEPT? I will wait for a minute and then I will check the chat box. Administration of an antibody linked to an enzyme such that the enzyme activates a prodrug. It is a very important therapy. Let me check the chat box now. One second. Yeah. Okay, so I have got only one answer which says it is number C. Okay, let me check if number C is the correct answer or not. So the correct answer for problem number 3 is yes, it is number C, ADEPT. Now let us discuss what is ADEPT. So basically, AD, the full form of the term ADEPT is the antibody directed enzyme prodrug therapy. Okay, so antibody directed enzyme prodrug therapy involves two steps. The first is the administration of an antibody enzyme complex. Okay, and the antibody is raised against tumor selective antigen and is linked to an enzyme such as bacterial carboxypeptidase. This complex then gets bound to the tumor. Unlike antibody drug conjugates, the antibody enzyme complex needs to remain attached to the surface of the cell and should not be internalized. A certain period of the time needs to elapse to give the complex time to bind the target cells or for unbound complex to be cleared from the blood supply. A prodrug of the cytotoxic drug is then administered. The prodrug is designed such it will be stable in the blood supply and can only be cleaved and activated by the enzyme complex to the antibody. This means that the toxic drug is only produced at the tumor and can be administered at a higher dose of the parent drug. CGS 149 is an example by which example of such product which is activated by the bacterial carboxypeptidase. An advantage of ADEPT over antibody drug conjugates is that the enzyme is catalytic and can generate a large number of active drug molecules at the site of the tumor. This can be diffused into tumor and affect the cells which are not have any antibody attached to them. A lot of research has been carried out on the ADEPT using bacterial enzymes such as carboxypeptidase G2, penicillin C acylase, and beta-lactamase. The advantage of using the foreign enzyme is that the enzymes can be chosen that are not present in the mammalian cell, but so there is no chance of the prodrug being activated by the mammalian enzyme during its circulation around the body. It is also possible to use foreign enzymes which have counterparts in the body as long as the latter are only present at the low level of blood supply or they are structurally distinct. Prodrugs can be designed that react selectively with the foreign enzyme rather than the mammalian version. Examples of enzymes in the category include beta-glucuronidase and nitroreductase. Many studies has been carried out on ADAPT. One example is an antibody beta-lactamase complex capable of reacting with the cephalosporin prodrug of the alkylating agent. So this is the example. Okay, this takes an advantage of the mechanism by the cephalosporin react to the beta-lactamase to eliminate the leaving group. So this is the beta-lactamase and this is the prodrug here, and this is the inactive alkylating agent. So ADP strategy follows here. So then the beta-lactamase, there is a, as you can see, there is a OH group which does this nucleophilic attack on the cephalosporin kind of group, and we have this active alkylating agent which is released. Okay, so this is a very good example of the carried out by ADPT strategy. Okay, so number C was the correct answer. Now I will go to the next problem. Problem number 4 says identify the incorrect statement. Your options are number A, ester products are used to mask the carboxylic acid functional group. Number B, enaraprilate is a prodrug for enarapril. Number C, ester products should be made moderately stable. Number D, it is hydrolyzed by the esterases to an alcohol and carboxylic acid which are quickly excreted due to the increased polarity. Which of the following statements is incorrect? So the statement says regarding the ester products, okay, and you have to identify the incorrect statement of the among the four. The first statement says that ester products are used to mask the carboxylic acid functional group. Number B says that enaraprilate is a prodrug of enarapril. Number C, ester products should be made moderately stable. Number D, it is hydrolyzed by esterases to an alcohol and carboxylic acid, which are quickly excreted from the body due to the increased polarity. Identify the incorrect statement. I will wait for 2 minutes, then I will check the chat box. Okay. Again, I am saying all of you try to answer the questions. Even if your answer is wrong, it's fine. But you should try to answer the question during the class. So which of the following statement is incorrect? Let me check the chat box. Okay, I have again only one answer, which is saying it is number C. Let me check if number C is the correct answer of the problem number 4 or not. So the statement which is incorrect is it is number B, which says that enalaprilate is a prodrug of enalapril. So basically, it is the other way around. Okay. So basically, enalapril is the prodrug and the active drug is the enalaprilate. Okay, so this enalapril is a ester prodrug for enalaprilate, which is the active drug. As you can see, this is the structure. This is the structure of enalapril, where R is the ethyl. As you can see, it is a ester functional group is there. Enalaprilate is the active form of this prodrug enalapril, which has a carboxylic acid group here. Okay, so basically, prodrug have been proved to be very useful in the temporary masking the awkward functional group, which is important to target binding but which hinders the drug from crossing the cell membranes of the gut wall. For example, a carboxylic acid functional group may have an important role to play in binding of the drug to its binding site via ionic and hydrogen bonding. However, it is very fact that it is an ionizable group may prevent it from crossing the fatty cell membrane. The answer is to protect the acid functional group as ester. The less polar ester can cross the fatty cell membrane, but once it is in bloodstream, it is hydrolyzed back to the free acid by esterases in the blood. For example, the extra product which is aid the membrane permeability is the enalapril. So it is a prodrug for the antihypertensive agent enalaprilate and pivamycin, which is a penicillin prodrug. So not all esters are hydrolyzed equally efficiently, and a range of esters may be need to try out to find the best one. Now, it is possible to make esters more susceptible to hydrolysis by introducing the electron withdrawing group of the alcohol moieties, such as OCH to CF3 or OCH to COOR or OCOCNH2 or OAR. Okay, the inductive effect of this group aids the hydrolytic mechanism by stabilizing the alkoxide leaving group, and care has to be taken, however, not to make the ester too reactive in case it has become chemically unstable, then hydrolyzed by the acid condition of the stomach or the more alkaline condition in the intestine before it reaches to the blood supply. To the end, it may be necessary to make the ester more stable. For example, cyclopropane carboxylic acid, this one esters, carboxylic acid esters have been studied for the potential product because the cyclopropane ring has the ability to stabilize the carbonyl group by the of the neighboring ester. Okay, so this is the respect acting as a bioisoster of the double bond, and the conjugate double bond stabilizes the neighboring carbonyl group due to the interaction with the pi systems involved. It is proposed that the sigma bonds of the cyclopropane ring are oriented correctly to allow the hyperconjugation interaction, hyperconjugative interaction that has been similar stabilizing effect on the neighboring carbonyl group. The interaction proposed involves the hyperconjugative donation to the antibonding pi orbital of the carbonyl group. And these are the other examples of the ester functional group, and also the thing. So there is a candoxatril. So this is also a very good example of the ester product, which has this 5-indenyl group as the ester group, and this is the active drug, which is the candoxatril. Okay. So let us review the statements again. So the first statement says that ester products are used to mark the carboxylic acid functional group. It is correct. Number B is false, which I have already mentioned that enalapril is the prodrug of enalapril, not the other way around. And number C says that ester product should be made moderately stable. It is also correct. It should not be very much stable, or not very highly stable or not very poorly stable. It should be moderately stable, so that it can cross the cell membrane and reach to the bloodstream. And when it is reaching to the bloodstream, it should be hydrolyzed by the esterases to release the active drug. And number D, it is hydrolyzed to an alcohol and carboxylic acid, which is also correct. Okay. So statement number B was false. Now let us go to the next problem. Problem number 5 says which functional group ensures the efficient conversion of 6-mercaptopurine to azathioprine. Your options are number A nitro, number B COOH, number C NH2, number D bromine or bromide. Which of the functional group ensures that efficient conversion of 6-mercaptopurine from azathioprine? NO2, COOH, NH2 or Br? I will give you 2 minutes, then I will check the chat box. Let me know. Meanwhile, A, B, C, or D, which of the following you think is the correct answer, or which of the following is the correct functional group, which ensures that the efficient conversion of 6-mercaptopurine from azathioprine. Which functional group ensures the efficient conversion of 6-mercaptopurine from azathioprine? Nitro, COOH, NH2, or bromo? Let me check the chat box. Okay, I have one answer which is saying it is number C. Okay, let me check if number C is the correct answer or not. So the functional group will be is nitro. The number A is the correct answer. Okay, and NO2 is the functional group which ensures the efficient conversion of the 6-mercaptopurine from azathioprine. So basically, this is the azathioprine. This is the structure of azathioprine. So as you can see, there is the purine ring here present, and along with that, we have this imidazole group here, which has a nitro, and this acts as a prodrug of the 6-mercaptopurine. Okay, so azathioprine is a prodrug of 6-mercaptopurine, and this is the how this particular conversion happens. So basically, sometimes the prodrugs are designed to be converted slowly to the active drug, thus prolonging the drug's activity. For example, 6-mercaptopurine, this is this one. So it suppresses the body's immune response and is therefore useful in protecting donor grafts. Okay, and unfortunately, the drug tends to be eliminated from the body too quickly. The prodrug azathioprine has the advantage that it is slowly converted to the 6-mercaptopurine by attacked by the glutathione. Okay, so glutathione is this one. Glutathione attacks it in here and then allowing a more sustainable activity. The rate of the conversion can be altered depending on the electron withdrawing ability of the heterocyclic group. The greater is the electron withdrawing power, faster is the breakdown. The nitro group is therefore present to ensure the efficient conversion of the 6-mercaptopurine as it is strongly electron withdrawing on the heterocyclic ring. Okay, so this nitro group is very much heterocyclic electron withdrawing due to this kind of resonance happens here on the nitro group. This group ensures that elimination or the conversion of this azathioprine to the active drug 6-mercaptopurine. Okay, and glutathione is here, which attacks basically on this position, and then the double bond goes here, and then the elimination happens. Okay, so this is a very good example how the prodrug is converted to the active molecule, active drug. So the correct answer was number A, nitro. Okay. So now I will go to the next problem. Problem number 6 says which amino acid residue is released from LDZ, a prodrug of diazepam. Your options are number A, valine, number B, lysine, number C, tyrosine, number D, alanine. Which amino acid residue is released from LDZ, which is a prodrug of diazepam? Valine, lysine, tyrosine, or alanine? Let me know A, B, C, or D which of the following is the correct answer, or which of the following is the correct amino acid residue released from LDZ, a prodrug from diazepam. So, which amino acid residue is released from LDZ? Let me check the chat box. Okay, so there is no answer. Okay, it is fine. Let me check which of the following will be the correct answer, and let us discuss it a little bit. So, the amino acid residue, which is basically released from LDZ, a prodrug of diazepam, will be, it is number B, lysine. So prodrugs masking the toxicity and side effects. So LDZ is an example of a diazepam prodrug which avoids the drowsiness side effects associated with the diazepam. Okay, so this is the structure of the LDZ. As you can see, there is a lysine side chain here. So basically, the side effects of associated with the high initial plasma levels of the diazepam followed by the administration. Okay, so prodrugs are not only always given to marks the to enhance the drug life or so on. It can be also given to avoid the side effects of the particular drug. Illicit is an excellent example for the same because it avoids the drowsiness side effect associated with the diazepam. The side effects are associated with the high initial plasma levels of diazepam following the administration. The use of the prodrugs avoids this problem. An aminopeptidase enzyme hydrolyzes the prodrug to release the non-toxin lysine moiety, and the resulting amine spontaneously cyclizes to the diazepam. So this is the structure of diazepam. So this is the structure of LDZ, which is the starting material, or this is the prodrug, and there is a lysine residue. If you can see, here is a lysine residue. And so first, this lysine residue is released, okay, and the enzyme which is responsible for the release of the lysine is the aminopeptidase enzyme. This is also which you need to remember, that aminopeptidase enzyme hydrolyzes this prodrug to form this I think this kind of active drug, and then what happens? When this active drug is formed, means there is a the cleavage happens, okay, and then this NH2 group is free, and this NH2 group acts as a nucleophile and attacks on this carbonyl carbon, and the cyclization happens, okay, followed by the proton elimination and addition. After that, we have this diazepam, which is our active drug. Okay, so because if we give diazepam directly, it will lead to the high initial plasma level, which will lead to the drowsiness. But if we give the LDZ, so it will release the diazepam little by little, which will give to the steady rise of the diazepam levels, not the initial high rise. So that is how this particular prodrug works. And here, this is an excellent thing that the amino acid side chain is used here, and the amino acid is the lysine, and the aminopeptidase enzyme helps this prodrug to get the convert to the active drug. Okay. Now I will go to the next problem. So problem number 7 says, which of the following statements is correct? Your options are: number A, palmitate ester of chloramphenicol increases its water solubility. Number B, palmitate ester of chloramphenicol is given intravenously. Number C, succinate ester of the chloramphenicol increases its water solubility. Number D, succinate ester of chloramphenicol is given orally. Which of the following statements is correct? Again, I will repeat. Number A, it says that palmitate ester of the chloramphenicol increases its water solubility. Number B, palmitate ester of chloramphenicol is given intravenously. Number C, succinate ester of chloramphenicol increases its water solubility. Number D, succinate ester of chloramphenicol is given orally. Which of the following statements is correct? Let me know A, B, C or D, which of the following statement you think is correct for problem number 7. I will wait for 1 or 2 minutes and then I will check the chat box. Even if your answer is wrong, it's fine, but please answer. Whatever you think is the correct answer, let me know. Number A, it says that palmitate ester of chloramphenicol increases its water solubility. Number B, palmitate ester of chloramphenicol is given intravenously. Number C, succinate ester of chloramphenicol increases its water solubility. And number D, succinate ester of chloramphenicol is given orally. Which will be correct answer? Let me check the chat box now. Okay, so there is no answer till now. Okay, it's fine. Let me check which of the following will be the correct answer for problem number 7. So the correct answer for problem number 7 will be, it is number C. Succinate ester of chloramphenicol increases its water solubility. This statement is true among these 4 options. Okay, so let us discuss it. So basically, this is the structure of the chloramphenicol. Okay, so chloramphenicol will be, there will be OH, but the chloramphenicol prodrugs like chloramphenicol palmitate. So this is the structure of the palmitate ester, which is where R is the CO, CH2 whole 14 CH3. And chloramphenicol succinate is the CO, CH2 whole 2 COOH. Okay, so prodrugs to lower the water solubility and prodrugs to improve the water solubility. The chloramphenicol prodrugs both have the example for the same. Okay, so some drugs have a revolting taste. One way to avoid this problem is to reduce their water solubility to prevent them dissolving on the tongue. For example, bitter taste of the antibiotic chloramphenicol can be avoided by using the palmitate ester. Okay, this is more hydrophobic because of the mixed alcohol group. The long fatty chain is present here. It is not. It does not dissolve easily on the tongue, but quickly hydrolyzed once swallowed. Okay, so the statement number 1, which says that palmitate ester of chloramphenicol increases the water solubility, it is false. Also, number B is incorrect, which says that it is given intravenously. No, the palmitate ester of the chloramphenicol actually decreases its water solubility so that it can be given orally. And when it is given orally, it does not get solubilized in the tongue because it has a bitter taste. So both first A and B statements are false. So then it says that prodrugs have been used to increase water solubility. Okay, this is particularly useful for the drugs which are given intravenously, as it means that the higher concentration and the smaller volumes can be used. For example, succinate ester of chloramphenicol increases letters water solubility because an extra carboxylic acid is present. Once the ester is hydrolyzed, chloramphenicol is released along with the succinic acid, which is naturally present in the body. Okay, so succinate ester, now the chloramphenicol increases its water solubility and it is quickly is dissolved in the quickly released from the in form of succinic acid in the body, which is naturally present in the body. So the R group is not toxic at all. And also it number D statement, which says that it is given orally, it is false because the succinate ester is given intravenously. So in short, the palmitate ester of chloramphenicol, it is decreases its water solubility and it is given orally, and succinate ester of chloramphenicol, it is given intravenously, and it in this ester, particular ester, increases its water solubility. Okay. So statement number C is the correct one for the problem number 7. Now I will go to the next problem. Problem number 8 says, which of the following is the correct reason for using hetacillin as the prodrug of ampicillin? Your options are: number A, increasing water solubility; number B, decreasing water solubility; number C, eliminating toxic metabolites; and number D, increasing chemical stability. Which of the following is the correct reason for using hetacillin as a prodrug of ampicillin? Number A, increasing water solubility; number B, decreasing water solubility; number C, eliminating toxic metabolites; and number D, increasing chemical stability. Let me know A, B, C or D, which of the following is the correct reason for using hetacillin as a prodrug of ampicillin. Why hetacillin is used as a prodrug of ampicillin? Because of the increasing water solubility, decreasing water solubility, eliminating toxic metabolites, and increasing chemical stability. Which will be the correct reason? Which of the following will be the correct reason for using hetacillin as the prodrug of ampicillin? Let me check the chat box now. Okay, so I have two answers. One is number C and another is number B. Let me check C or B, which of the following will be the correct answer for problem number 8. So the correct answer will be, it is number D, increasing chemical stability. So prodrugs to increase the chemical stability is another example of the prodrug hetacillin. So basically this is the structure of the hetacillin and this is the structure of the ampicillin. Okay, and the antibacterial agent ampicillin decomposes in concentrated aqueous solution, and as a result of the intramolecular attack for the side chain of amino acid of the lactam ring. Okay, hetacillin is a prodrug which locks up the offending nitrogen into the inner ring. Okay, and it prevents the reaction. Once the product has an has been administered, the heterocillin slowly decomposes to release the ampicillin and acetone. In the field of antiviral agents, cyclopropane carboxylic acid ester are also being studied for the potential product of acyclovir in order to prolong the chemical stability of the solution. Okay, so this is the reason why ethacillin is used as a product of ampicillin for to increase the chemical stability, because there is a nitrogen NH2 group here, as you can see. So this basically this nitrogen, it involves in the at intermolecular attack of the side chain of amino acid group of the lepton ring. This one, okay, and hetacillin is a product which has this nitrogen locked in this in a ring. Okay, and because of this locked position, it is stable more, and when it is released, the ampicillin is released, there is acetone which is released from this position. Okay, and so this is the very good example where product is used to increase the chemical stability. Okay, so option number D was the correct answer for problem number 8. Now I will go to the next problem. Problem number 9 says molecules which are formed by reducing the peptide nature in a lead molecule is known as. Your options are number A peptidomimetic, number B oligonucleotide, number C prototype, and number D neurotransmitter. Molecules which are formed by reducing the peptide nature in the lead molecule is known as dash. You have to fill in the blanks. Your options are number A peptidomimetic, number B oligonucleotide, number C prototype, number D neurotransmitter. What are these molecules called which are formed by the reducing the peptide nature in the lead molecule? Peptidomimetic, oligonucleotide, prototype, or neurotransmitter? Let me know. I will wait for some time and then I will check the chat box. Hello. I hope I am audible. If there is any issue with the audio, please let me know. So my voice is audible, right? Can anybody confirm in the chat box if it is audible or not? Hello. So I am audible or not? Please let me know in the chat box if the voice is audible. Anyone? Anyone present in the class, please can you confirm that if my voice is audible or is it not? Hello. Okay, fine. Please, time to time, let me know if there is any issue with the or sound or anything. Okay. So, okay, I have got the answer which says it is number A for the problem number 9, I guess. So let me check if number A is the correct answer for problem number 9. So the molecules which are formed by reducing the peptide nature of the lead molecule is known as Yes, number A is the correct answer, peptidomimetic. Okay, peptidomimetic is the correct answer. So now let us discuss what is peptidomimetic. So peptidomimetics incorporating the conformational constraint logically or globally. What is peptidomimetics? Let us discuss. So these are the molecules with significantly reduced peptide character that mimic the bioactive conformation of the peptides and thus retain the ability to interact with the biological target and cause the same biological effect. Small protein-like chain designed to mimic a peptide arise either from the modification of an existing peptide or by designing the similar systems to mimic the peptides. For example, peptides or beta peptides. Okay, so peptidomimetics are molecules which look or feel like behave like peptides. Okay, so because peptides continue to be important lead compounds, there are medicinal chemistry has worked around the peptides, and because peptides are receptor and ligands and enzyme substrates are very commonly found. Therefore, we do not want to give up on the peptides altogether. So therefore, we want to be able to deliver the peptide or the peptide-like molecule, which is known as the peptidomimetic. So consequently, drugs which are designed from these lead compounds, which are peptide-like in nature, have been taken forward. So the peptidokinetic properties, so the pharmacokinetic properties of this first generation drugs using the peptides are many times unsatisfactory. Okay, and so a number of strategies has been evolved to improve the bioavailability and to achieve a higher level or acceptable level of these molecules in the blood supply. So therefore, disguising or reducing the peptide nature of the lead compound to generate a structure which is more easily absorbed from the GI tract and more resistant to digestive and metabolic enzymes is highly desirable, and these molecules are known as the peptidomimetics. Okay, we will discuss regarding this peptidomimetic more in our today's class and on the upcoming classes also. So yes, the correct answer was peptidomimetic. Okay, so yeah, number A was the correct answer. And in the chat box, I also received it number A. So very good. So now let us move to the next problem. Problem number 10 says, which of the following is a peptidomimetic formation technique? Your options are number A, pegylation, number B, replacing L amino acid with D, number C, N-methylation, number D, all of the above. Which of the following is a peptidomimetic formation technique? Number A is the pegylation, number B, replacing L amino acid with D, number C, N-methylation, and number D, all of the above. Which of the following is a peptidomimetic formation technique? I will wait for 1 or 2 minutes, then I will check the chat box. Okay. Pegylation, replacing L amino acid with D, N-methylation, or all of the above. Which is our peptidomimetic formation technique? Which of the following will be peptidomimetic formation technique? Let me know in the chat box. Okay, let me check the chat box first. Okay, I have got one answer which saying it is number D. Let me check if number D is the correct answer or not. So the correct answer will be yes, it is number D. All of the above techniques are the peptidomimetic formation techniques, pegylation, replacing L amino acid with D, and L-methylation. So peptidomimetics as drug, disguising or reducing the peptide nature of the lead compound to generate a structure which is more easily absorbed from the gastrointestinal tract and is more resistant to the digestive and metabolic enzymes. Such analogues are known as the peptidomimetics. It has been found that the linking of the polymer polyethylene glycol or PEG is to a protein can increase lattice solubility and stability, as well as decreasing the likelihood of an immune response. Pegylation, as it is called, also prevents the removal of the small proteins from the blood supply by the kidneys or the reticuloendothelial system. The increased size of the pegylated protein means that it is not filtered into the kidney nephrons and remains in the blood supply. The PEG molecule surrounding the protein can be viewed as a kind of the hydrophilic and polymeric shield which both protects and disguises the protein. The PEG polymer has been added advantage that it shows little toxicity. So the methyl group acts as a steric shield and prevents the important hydrogen bonding interaction that is supposed to take place between the NH of the original amide and the active site of the peptidase enzyme. So therefore, now the steric shield is present, the rates of the hydrolysis significantly go down. So in-methylation also is a one way to generate the amide-like or to retain the properties of amide as a drug. Another unrelated or a completely different strategy is to flip the enantiomer as we use. So for example, if we replace the N-enantiomer that is normally present in the proteins with the D-enantiomer, then what happens that it makes the relative orientation unrecognizable to the digestive or metabolic enzymes, especially if the side chain is involved in binding interaction. So there's an example, if you have a L amino acid and if you convert, you will have two L amino acids, but if you convert one to the D amino acid of the two, now this to the D amino acid, it is possible that it may not be hydrolyzed by the original molecule. So pegylation, replacing L amino acid with D, and in methylation, all these are the techniques for the formation of the peptidomimetic. Okay. So number D was the correct answer. Now let me go to the next problem. Problem number 11 says, which enzyme is responsible for SCID, which is the severe combined immunodeficiency syndrome? Okay, severe combined immunodeficiency syndrome, which enzyme is responsible for the same? Options are number A, catechol dehydrogenase, number B, cytochrome C oxidase, number C, adenosine deaminase, number D, adenylate deaminase. Which enzyme is responsible for SCID, severe combined immunodeficiency syndrome? Number A, catechol dehydrogenase. Number B, cytochrome C oxidase. Number C, adenosine deaminase. And number D, adenylate deaminase. A, B, C or D, which of the following is the correct answer? Which enzyme is responsible for this particular disease, which is the severe combined immunodeficiency syndrome or SCID? Your options are number A, catechol dehydrogenase, number B, cytochrome C oxidase, number C, adenosine deaminase, and number D, adenylate deaminase. I will wait for 2 to 3 minutes, then I will check the chat box, and after that, I will show you what is the correct answer and we will discuss it a little bit. So, which of the following enzymes are responsible for SCID? Catechol dehydrogenase, cytochrome C oxidase, adenosine deaminase, or adenylate deaminase. A, B, C, or D, which of the following will be the correct answer? Let me check the chat box. Okay, I don't have any answer for problem number 11. Okay, it's fine. Let me show you the correct answer and let us discuss it a little bit. So the enzyme which is responsible is number C, adenosine deaminase. This adenosine deaminase is the enzyme which is responsible for SCID. So What does this adenosine deaminase do? Basically, this is the adenosine and this is the inosine. So as you can see, adenosine dehydrogenase convert this adenosine to the inosine. Okay. So SCID or severe combined immunodeficiencies consists of a group of genetic disorders characterized by a block in T lymphocyte differentiation that is variably associated with the abnormal development of other lymphocyte lineages, that is B or NK lymphocyte, and more rarely of the myeloid lineage. A genetic disorder in which both arms of the B cells and T cells of the adaptive immune system are impaired due to the defect in one of the several possible genes. SCID is a severe form of the heritable immunodeficiency. The overall frequency is estimated to be 1 in the 75,000 to 1 lakh births. So, PEG molecules surrounding the protein can be viewed as a kind of hydrophilic polymeric shield which both protects and disguises the protein. The PEG polymer has been added advantage that it shows little toxicity. The enzyme L-asparaginase and adenosine deaminase have been treated in this way to give the protein PEG conjugate called pegasparase and pegadrimase, also which have been used for the treatment of the leukemia and severe combined immunodeficiency or SCID syndrome specifically, respectively. SCID is an immunological defect associated with the lack of the adenosine deaminase. The conjugates have been longer plasma half-life than the enzyme alone and are less likely to produce an immune response. Interferon has similarly been pegylated to give the preparation known as the peginterferon alpha to B, which is used to treatment of hepatitis C. Okay. Here it is also an example how this product pegylation can help in combining the SCID. Okay, because this SCID is generated with the in this due to the lack of this particular enzyme, and the conjugates have the longer, the PEG conjugate of this enzymes have the longer half life, which is than the enzyme alone and less likely to produce an immune response. Okay, so number C was the correct answer. Now I will go to the next problem. Problem number 12 says, which of the following is not an example of the peptide drug? Your options are number A, cyclosporine, number B, enfuvirtide, number C, teriparatide, and number D, artemisinin. Which of the following is not an example of the peptide drug? Cyclosporine, enfuvirtide, teriparatide, or artemisinin? A, B, C, or D, let me know which of the following is not an example of peptide drug. Cyclosporine, enfuvirtide, teriparatide, or artemisinin. Which will be the correct answer? I will wait for 2 to 3 minutes and then I will check the chat box. Which of the following will not be an example of the peptide drug? মা কথা বলো না। Hello. Am I audible? Which of the following is not an example of peptide drug? Cyclosporine, enfuvirtide, teriparatide, or artemisinin? Let me check the chat box now. Okay, I have got only one answer, which is number B. Let me check if number B is the correct answer or not. Okay, so the correct answer for problem number 12 will be, it's number D, artemisinin. Artemisinin is not an example for the peptide drug, but the first three are the example of the peptide drug. So this is the structure of the cyclosporine. This is the structure of the enfovertide. This is the structure for the teriparatide, and this is the structure for artemisinin. So from the structures, it is clearly you can see all the first three, the cyclosporine, enfovertide, and teriparatide are the peptide drugs, but not the artemisinin. It is a normal compound. It is not. There is no peptide here. Okay, so as the structures, you can show the peptide drugs that we have a very complex structure. You don't need to memorize the structure at all, but yeah, you just need to remember the names of the particular examples of the peptide drugs. So often, there is often reluctance to use the peptides as the drugs because there are many pharmacokinetic difficulties that can be encountered. But that does not mean that the peptide drugs have no role to play in the medicinal chemistry. For example, the immunosuppressant cyclosporine can be administered orally. This is an important peptide drug is goserelin, which is administered as a subcutaneous implant and is used against the breast and prostate cancer, earning $700 million per year as a maker. In 2003, enfuvatide, or it is has a marketed name of Fuzenon, was approved as the first new class of anti-HIV drugs. In the polypeptide, it is a polypeptide of 36 amino acids which are injected subcutaneously and offers another weapon to combination therapies and used against HIV. Teriparatide, which is also mentioned, is also administered by subcutaneous injection. Peptide drugs can be useful if one chooses the right disease and the method of administration. Okay, so there are although there are various pharmacokinetic difficulties, because as you remember, very important rule which we have discussed till now, that the rule of the Lipinski's rule and so on, which says that the drug molecule should be have lower molecular weight and so on, so things. So very good example was cyclosporine. I have already mentioned it then also when we was discussing about the I think the Lipinski's rule, that the cyclosporine does not follow the Lipinski's rule, but still it can be administered orally, whereas these drugs are administered in the subcutaneous injections, and all these are peptide drugs. Artemisinin is not a peptide drug. It is a basically it is separated as a natural product from a plant. Okay, this is a very good natural product, and this is used as an anti-malarial agent. Okay, so number D was the correct answer for problem number 12. Now let me go to the next problem. So problem number 13 says, which of the following statements regarding neurotransmitters are is not true? Your options are: number A, they are secreted by neuronal cell. Number B, they are chemical messengers. Number C, they basically traverse through the blood to act on the distant target cell. Number D, they carry out the cascade of reactions. Which of the following statements regarding neurotransmitters is not true? Read all the options very carefully, then let me know in the chat box A, B, C or D which of the following statement is the correct answer for problem number 13. You have to basically identify the incorrect statement regarding the neurotransmitters. Your options are: number A, they are secreted by the neuronal cells. Number B, they are chemical messengers. Number C, they basically traverse through the blood to act on a distant target cells. Number D, they carry out cascade of reactions. Which of the following statements regarding neurotransmitter is not true? I will wait for a minute, then I will check the chat box and we will discuss that particular question and answer. Okay. Let me check the chat box. Okay. I have got one answer. I think it is for problem number 13. Yes, which says it is number C. Okay, let me check if number C is the correct answer. So the correct answer means the statement which is not true regarding neurotransmitters will be, yes, it is number C. They basically traverse through the blood to act on a distant target cell. So neurotransmitters, so the definition says that they are the chemical substances released by the electrical impulses into the synaptic cleft from the synaptic vesicles of presynaptic membrane. It then diffuses to the prosynaptic membrane, binds to and activates the receptors present, leading to the initiation of the new electrical signals or inhibition of the postsynaptic neuron. The examples of the neurotransmitters are the adrenaline, which is known as the fight or flight, which is produced in the stressful situation, increases the heart rate and blood flow, leading to the physical boost and heightened awareness. GABA, which is the gamma aminobutyric acid, which has the calming effect of the coming the comes the firing nerves of the CNS, and high levels to improve focus, low levels cause anxiety. Also contributes to the motor control and vision. We have the noradrenaline, which gives the concentration, affects the attention and responding the actions in the brain, contracts the blood vessels, increasing blood flow. Acetylcholine is for learning, involved in thought, learning and memory, activates the muscle action in the body, also associated with the attention and awakening. Dopamine for pleasure, feeling of the pleasure, auto addiction, also addiction, movement and motivation. People repeat the behaviors that lead to the dopamine release. Glutamate for memory, most common neurotransmitter involved in learning and memory, regulates the development and creation of the nerve cell. Serotonin for the mood, it contributes to the well-being and happiness, helps to sleep cycle, digestive system regulation affected by the exercise and light exposure, and endorphins, which gives the euphoria and which released during exercise and excitement, producing well-being and euphoria and reducing the pain. Okay, so these are the examples of the neurotransmitter along with what the particular neurotransmitter do. So there is another very important thing which is known as the hormones. So hormone and neurotransmitter, they are sometimes they seem to be very similar, but they have certain differences. Like hormones are the released by the endocrine glands, whereas the neurotransmitters are released by the nerve cells. Hormone travel through the blood and act on the distant target cell, but neurotransmitter travel across the synapse to act on the nearby cells. The effects of the hormone can be slower to build but longer lasting. The effects of the neurotransmitter can be fast to act but shorter lasting. Hormones are a part of the endocrine system, neurotransmitter are part of the nervous system. So the statement number 1, which says that neurotransmitter secreted by neuronal cell, it is correct. They are chemical messenger, it is also correct. They basically traverse through blood to act on a distant target cell. This is not correct. This is the incorrect statement. They act. They traverse. They actually act on the nearby cells. Okay, and they carry out a cascade of reaction. It is also correct. So next statement number C, it was false regarding the neurotransmitter. Now I will go to the next problem. Problem number 14 says megastrenol acetate has a short duration of activity since it is metabolized at the position 6. Here, okay, placing a methyl group at 6th position increases the duration of the action. What role does the methyl group play? Number A, it is a susceptible metabolic group. Number B, it is a metabolic blocker. Number C, it is a conformational blocker. Number D, it is an electron donating group. Again, I will repeat problem number 14. Magistral acetate, which is this one. This is the structure of the magistral acetate, and has a short duration of activity since it is metabolized at the problem number 6. Sorry, it is the position number 6. So here on the 6th position, it is metabolized. Here it is marked here. Placing a methyl group here in the 6th position increases the duration of the action. What role does the methyl group play? If there is a methyl group here, it will increase the duration of action. What does this methyl role play? Number A, it is susceptible metabolic group. Number B, it is a metabolic blocker. Number C, it is a conformational blocker. Number D, it is an electron donating group. Which of the following will be the correct answer? A, B, C or D? Let me know in the chat box. If we place a methyl group on the 6th position to increase the duration of action for magistral acetate, what role does this methyl group play? As a susceptible metabolic group, as a metabolic blocker, as a conformational blocker, or as a electron donating group? So let me check the chat box. Okay, I think I have received one answer which says it is number C. Let me check if number C is the correct answer for the problem number 14. So, if we introduce a methyl group on the 6th position of magistral acetate, the role of this methyl group will be number B. It is a metabolic blocker. So, some drugs are metabolized by the introduction of the polar groups at a particular position in the skeleton. For example, steroids can be oxidized at the position 6 of the tetracyclic ring to introduce a polar hydroxyl group. The introduction of this group allows the formation of the polar conjugates which can be eliminated quickly from the system. By introducing a methyl group on the position number 6 is metabolized. Position number 6, this metabolism is blocked and the activity of the steroids is prolonged. Okay, the oral contraceptive magistral acetate is an agent which contains the 6-methyl blocking group. On the same lines, the particular method of protecting aromatic ring is the from metabolism is the para position to introduce the fluorosubstituent. For example, the CGP 52411, which is this compound, is an enzyme inhibitor which acts on the kinase active site of the epidermal growth factor receptor or EGFR. It went forward in the clinical trials as an anti-cancer agent and was found to undergo the oxidative metabolism at the para position of the aromatic figures. Fluorosubstituents were successfully added to the analog CGP 53353 to block the metabolism. This tactic was also applied successfully to the design of the gefitinib. Fluorine was now part of the used extensively to block the metabolism of a various structural substitution. Another approach is to actively being exposed is which is actively being explored is the replace of the hydrogen atom to the deuterium isotope. The covalent bond between the carbon and deuterium is twice as strong as between carbon and hydrogen, and this might help to block the metabolic mechanisms. Okay, so this is the stearl acetate which I have already mentioned, and so this is the susceptible. So there's this side is the for metabolic oxidation, and if we introduce a methyl here, it will act as a metabolic blocker. Now, in place of methyl, we can also use fluorine, as I have already mentioned here. The use of fluorine substitute as a metabolic blocker in this particular agent, which is used as a kinase inhibitor. Okay, so the correct answer was problem number B. Now I will go to the next problem. Problem number 15 says oligonucleotides suffer disadvantages as drugs since they are susceptible to the enzyme catalase degradation. Which of the enzyme is mainly responsible for this? Your options are number A peptidases, number B nucleases, number C lipases, and number D oxidases. Oligonucleotides suffer disadvantages as the drug since they are susceptible to enzyme catalase degradation. Which of the following enzyme is mainly responsible for this? Peptidases, nucleases, lipases, or oxidases? Let me know A, B, C, or D. Which of the following you think is the correct answer for problem number 15? Which enzyme is responsible for the degradation for the oligonucleotides? And which enzymes are responsible for the disadvantages of these oligonucleotides as a drug? Peptidases, nucleases, lipases, or oxidases? I will wait for a minute or two, then I will check the chat box. Okay. Which of the following enzymes is responsible for the disadvantages of oligonucleotides as a drug? Peptidases, nucleases, lipases, or oxidases? I wait for a minute, then I will check the chat box. Okay, let me check the chat box now. Okay, I have one answer which says it is number B. Okay, let me check if number B is the correct answer for problem number 15. The correct answer will be, yes, it is number B, nucleases. So, oligonucleotides as the drug. So, oligonucleotides are being studied as the antisense drugs and aptamers. The rationally and the therapeutic potential of these agents is described here. So, however, there are disadvantages for the use of oligonucleotide as a drug as they are rapidly degraded by the enzymes called nucleases. They are also large and highly charged and are not easily absorbed through the cell membranes. Attempts to stabilize these molecules and to reduce their polarity have involved modifying the phosphate linkage in the sugar phosphate backbone. For example, phosphorothiones and the methyl phosphonates have been extensively studied, and oligonucleotides containing these linkages show promise as a therapeutic agents. An antisense oligonucleotide with such a modified backbone has been approved as an antiviral drug. Alterations to the sugar moiety have been also tried. For example, placing the methoxy group at the position number 2 to prime or the using of alpha anomer of the deoxyribose sugar increases the resistance to nucleases. Bases have also been modified to increase the number of the hydrogen bonding interaction with the target nucleic acids. The biopharmaceutical company Genta has developed an antisense drug called oblimersen, which consists of the 18 deoxynucleotides linked to the phosphorylate backbone. It also binds the initiation codon for the messenger RNA and molecule carrying the genetic instruction for BCL2. BCL2 is a protein which suppresses the cell death or apoptosis, and also so suppressing the synthesis will also increase the chances for the apoptosis taking place in the when chemotherapy or radiotherapy is being used as a treatment of cancer. The drug is currently undergoing the phase 3 clinical trials in combination with the anti-cancer drug docetaxel and irinotecan. So phosphothioate oligonucleotides also being investigated, which will target the genetic instruction for the RAF and PKC gamma, two proteins which are involved in the signal transduction pathway. These are two have potential as anti-cancer drugs. So these are the modifications on oligonucleotides to increase its stability and use as a drug. Okay. So yeah, number B was the correct answer. Nucleases are the enzymes which basically degrades this oligonucleotides, hence limiting their use as a drugs. Now I will go to the next problem. Problem number 16 says cyclophosphoramide is commonly used in anti-cancer therapy. Which of the following statements is not true of the its structure? Number A, it is relatively non-toxic. Number B, it acts as a prodrug. Number C, it cannot be taken orally. Number D, the structure is metabolized to release acrolein. Cyclophosphamide is commonly used as anti-cancer therapy. Which of the following statements is not true about its structure? Number A, it is relatively non-toxic. Number B, it acts as a prodrug. Number C, it cannot be taken orally. Number D, the structure is metabolized to release acrolein. A, B, C or D, which of the following will be the correct answer for problem number 16? I will wait for 1 or 2 minutes and then I will check the chat box. Which of the following will be the incorrect statement regarding the cyclophosphamide? Let me check the chat box. Okay, there is no answer for the problem. Okay, let me check which of the following will be the correct answer for problem number 16. So the correct answer will be number C. It cannot be taken orally. This statement is not true regarding the cyclophosphamide. So this is the cyclophosphamide. Already I have discussed it a couple of times that it is a very good alkylating agent. So this is the structure of cyclophosphamide and CYP 450 family of enzymes. They form this 4-OE cyclophosphamide and this then aldophosphamide, which goes to give rise to the phosphorite mustard, which is the DNA protein and alkylation. So this induces the cell death, and also we have the acrolein, which is the give rise to to the tissue toxicity. The cyclophosphamide is a successful non-toxic prodrug which can be taken orally. Then once absorbed, it is metabolized in liver to the toxic alkylating agent which is useful for the treatment of cancer. Okay, so statement number C, which says that it cannot be taken orally, it is incorrect. It is a prodrug and it can be taken orally. Now let us go to the next problem. Problem number 70 says, why should the addition of an alcohol or phenol group to the drug decrease its duration of action? Your options are: number A, it acts as a polar handle for conjugate reaction. The conjugates are excreted more quickly. Number B, it increases the polarity of the drug and reduces the amount of drug absorbed. Number C, it reacts with the proteins in the body such that the drug is irreversibly linked to the other proteins by covalent bond. Number D, it acts as an electron withdrawing group and affects the binding strength of the important binding groups. Identify the correct answer for problem number 17, which says, why should the addition of an alcohol and phenol group to the drug decreases its duration of action? Your options are: number A, it acts as a polar handle for conjugate reactions. The conjugates are excreted more quickly. Number B, it increases the polarity of the drug and reduces the amount of drug absorbed. Number C, it reacts with proteins in the body such that the drug is irreversibly linked to the proteins by covalent bond. Number D, it acts as an electron withdrawing group and affects the binding strength for the important binding groups. Why should the addition of an alcohol or phenol group to the drug decrease its duration of action? Which will be the correct answer? Number A, it acts as a polar handle for conjugation reaction. The conjugates are excreted more quickly. Number B, it increases the polarity of the drug and reduces the amount of the drug absorbed. Number C, it reacts with proteins in the body such that the drug is irreversibly linked to the proteins by covalent bond. Number D, it acts as an electron withdrawing group and affects the binding strength of the important binding groups. Which of the following will be the correct answer for problem number 17? I will wait for a minute, then I will check the chat box. So why should the addition of alcohol or phenol group to the drug decrease its duration of action? Let me check. Okay, there is no answer. Okay, fine. The correct answer is number A. It acts as a polar handle for the conjugation reaction. The conjugates are excreted more quickly. So basically, it is related to the phase 2 conjugation reactions, which I have already, if you mention, I have mentioned it a couple of times. So basically, the R group by in the lipophilic and hydrolysis reduction. So for one is the oxidation reaction where the oxygen is inserted like in form of ketone or epoxides, and we have the interaction of the nucleophiles. So these are the polar phase 1 and these are the phase 2 reaction, metabolic reaction. So in case of the phase 2, we have the this glutathione reaction and sulfation, acylation, and glucuronidation, and these are the following phase 2 conjugation reactions. So what are the following groups are involved? The sulfate, alpha glucuronidinic acid, glutathione, methyl, glycine, glutamine acyl. These groups are transferred by the appropriate conjugation enzymes on the drugs and other metabolites. Okay, so these things I have discussed in, I think, our week 6 or 7, where we were discussing the different phases of pharmacokinetics at the phase 1 and phase 2 metabolic reactions. So phase two conjugation reaction are basically at the as the formation of the different conjugates which can okay, so this is the that was the number A is the correct answer. Now let me go to the next problem. Problem number 18 says which of the following statement is incorrect. Your options are: number A, ticonazole can only be applied through skin. Number B, fluconazole is more polar than ticonazole. Number C, fluconazole has a better interaction with blood. And number D, ticonazole can be only applied intravenously. A, B, C or D, which of the following statement is incorrect? Number A, ticonazole can only be applied through skin. Number B, fluconazole is more polar than ticonazole. Number C, fluconazole has a better interaction with blood. Number D, ticonazole can be only applied intravenously. A, B, C or D, which of the following statement is incorrect? Again, I will repeat. Number A, ticonazole can only be applied through skin. Number B, fluconazole is more polar than ticonazole. Number C, fluconazole has a better interaction with blood. And number D, ticonazole can only be applied intravenously. Which of the following statement will be incorrect? I will wait for some time and then I will check the chat box and we will discuss this particular question. So, which of the following statement is incorrect? Let me check the chat box. Okay, I have one answer which says it is number D. Let me check if number D is the correct answer or not. So the statement which is incorrect, it is number D. Ticonazole can only be applied intravenously. This statement is incorrect. So this is the structure of the ticonazole. So basically ticonazole, fluconazole, all these are the azole containing antifungal agents. Okay, because as you can see in fluconazole we have two triazole rings here present. 1 to 4 triazole rings are present here, and ticonazole it has a imidazole ring which is present here. So azoles are a very good antifungal agents. So adding or removing polar functional groups to vary the polarity. So a polar functional group could be added to a drug to increase its polarity. For example, the antifungal agent ticonazole is only used for skin infection because it is non-polar and poorly soluble in blood. Okay, so ticonazole it is non-polar, it is not soluble in blood, so it can only be used topically on the skin infections as a form of the cream or ointment. Introducing a polar hydroxyl group and more polar heterocyclic rings lead to orally active antifungal agent fluconazole with improved solubility, enhanced activity against the systematic infection. So this is the fluconazole. So fluconazole is more polar than ticonazole because it has a polar group, hydroxyl group, and also triazole rings, which are more polar than the imidazole ring or any this kind of aromatic rings. Also, fluorines are present here. They also increases the polarity of the fluconazole. Since fluconazole is a more polar, and that's why it has improved solubility and enhanced activity. And the fluconazole can be used in case in the systematic infections, in the infection or the fungal infection which happens in our internal system or blood or anything. Okay, but ticonazole, since it has a poor solubility and it is non-polar, it cannot be used intravenously. It only can be used applied through skin. So the statement number A, which says that iconazole can be only applied through skin, is correct. Number B, which says that fluconazole is more polar than ticonazole, it is also correct. Number C, which says fluconazole has better interaction with blood, it is also correct. Number D is incorrect, which says that ticonazole can be only applied intravenously. No, ticonazole can be only used topically and form of the on the skin infection as the creams or in ointments. Okay. So yes, number D was the incorrect statement. Now, problem number 19. It says that there are several strategies for stabilizing susceptible bonds in peptides. Which of the following is not used in this respect? Number A, replace the peptide link with the stable functional group. Number B, adding an N-methyl group to the peptide link. Number C, using an unnatural amino acid. Number D, replacing D amino acid with L amino acid. There are several strategies for stabilizing the susceptible bonds in peptide. Now, which of the following is not used in this respect? Number A, replace the peptide link with a stable functional group. Number B, adding an N-methyl group to the peptide link. Number C, using an unnatural amino acid. And number D, replacing the D amino acid with L amino acid. Which of the following is not used as the strategies for stabilizing susceptible bonds in peptides? Number A, replace the peptide link with the stable functional group. Number B, adding N-methyl group to peptide link. Number C, using an unnatural amino acid. Number D, replacing D amino acid with L amino acid. Which of the following will be the correct answer for problem number 19? Let me check the chat box. So there is no answer for problem number 19. Okay, it's fine. Let me just check which of the following will be the correct answer. So the strategy which is not used to for stabilizing the susceptible bonds in peptide will be It is number D, replacing the D amino acid with L amino acid. Okay, so the replacing D amino acid with L amino acid, this strategy is not used for stabilizing the susceptible bonds in peptides. So what are the differences between the L amino acid and D amino acid and why? L amino acid is stereoisomer of the particular amino acid whose amine group on the left inside of the Fischer projection. And D amino acid is stereoisomer of the amino acid whose amino group is the right side of the Fischer projection. L amino acid can rotate the plane polarized light in counterclockwise in the process called lever rotation. D amino acid can rotate the plane polarized light clockwise in a process called dextrorotation. L amino acid is superseded by the R notation, and D amino acid they are superseded by the S notation. Now, L amino acid used by cell to produce protein, D amino acid occur in the cell wall of the bacteria. Okay. Other strategies, which first one which says that the replacing peptide link with stable functional group, it is used adding an N-methyl group to the peptide link. It is also used using unnatural amino acid. It is also first three strategies are used to stabilizing the susceptible bonds in peptide, but not the fourth one, like replacing D amino acid with L amino acid. Okay. Now, last question of today's session, it says why morpholine and pyridine rings are added to the drug molecules. Options are number A, to improve the hydrophobicity, number B, to improve the water solubility, number C, to increase the molecular weight, and number D, all of the above. Which of the following will be the correct reason of the usage of morpholine and pyridine links in the drug molecules? Number A, to improve the hydrophobicity, number B, to improve water solubility, number C, to increase the molecular weight, and number D, all of the above. Why morpholine and pyridine rings are added to the drug molecules? Why morpholine and pyridine rings are added to the drug molecule? Let me check the chat box. So I have got one answer which says it's number A. Let me check if number A is the correct answer or not. So the correct answer will be, it is number B, to improve the water solubility. This is the structure of morpholine, this is the structure of pyridine. So the first question explains why morpholine and pyridine are added to the drug molecule. So in many times what we have find that the drug molecule is actually quite poorly soluble, and so in order to improve the solubility one needs to add groups which can be hydrophilic in nature, but they are not going to dramatically alter the properties of the drug. So here nitrogen containing heterocycles such as morpholine and pyridine are quite often used. So the nitrogen is polar and definitely increases the polarity, and in some cases protonated form also is isolated as water soluble salts. So here there is an for example, if you connect it through the this nitrogen and then you have sort of this whole molecule is of this molecule. Morpholine is quite polar, and here in case pyridine, you can make pyridinium salt actually isolated as a pyridinium salt. So if a polar group is added in order to increase the solubility, it is preferable to add it to the molecule in such a way that it's still exposed to surrounding water. Okay, so therefore, because it is exposed to surrounding water, the drug is then bound to the target site, does not have any sort of the dissolvate before binding to the active site. So this is an important criterion when we are trying to add more functional group to the drug. Okay, so to improve water solubility, generally this pyridine and morpholine rings are added to the drug molecule. So this was the last question of today's session. So basically today what we did, we mainly discussed regarding the prodrug. We discussed different strategies for the prodrug formation, how prodrugs they affect the activity of the drug, why the prodrugs are used in different aspects, what are the designing method, what are the limitation, all these things we discussed along with the peptidomimetic strategies and neurotransmitter a little bit. So today it was our week 9 live session, and I will share my email ID with you. If you have any doubt or any query regarding today's class, you can let me know through this email ID. Okay. So this is my email ID in the chat box. If you have any doubt, any query in regarding today's class or any topic in general, you can let me know through the email. The class recording and also the presentation I will be uploading into the course page. So you know where is the course page. I have already mentioned. If you go to the SWAM portal in the medicinal chemistry, you will find there is a left panel where the weeks are updated, and there is a problem solving session recording. If you click on here, you will be redirected to this page, and clicking on this icon will be giving you all the class materials and also the recordings. Okay, so today it was our week 9 live session. I will if there is no doubts or queries, I will close the session for today. We will meet on the next Friday from this 4 to 6 p.m. class, and we will discuss in the week 10 course content in the next class. Okay, today it was our week 9 class, ninth class. So we discussed the week 9 course content. So is there any doubt or any problem regarding today's class? You can let me know now. Also, you can let me know through the email which I have shared to you here. So if there is no query, I will close the session for today. So thank you all for joining. We will meet on the next class, which will be on next Friday from 4 p.m. Okay. So I am closing the session for today. Thank you all.